Hello Panthers, we are back after exam day with the adjustment of our class online. Please don't forget to be strong, to be wise, to be disciplined, and to remain focused so that you can be successful. At this point, we have discussed two different classes of polymers of biomolecules. We have done the polymers of glucoses that we call starch, amylose, amylopectin, and also the molecules of cellulose. We have seen how carboxylic acid ends can combine with amino ends to make peptide bonds, making a polymer of amino acids that we call a protein. And in this chapter, we address how a nucleotide can make a polynucleotide that is going to be called a nucleic acid. So this is the chapter of the family of nucleic acids. And we know a little bit of what is called the molecule of DNA and RNA, and we're going to explore more of the details. The first thing is that we need to remember it's a molecule of DNA is a very long molecule. It will exist in the nucleus of the cell, and I have here the nucleus of the cell, as a compact, orderly, tangled DNA twisted molecule that is going to fit in the cell because it's surrounded, it's wrapped about proteins called histones. I have this picture here to show you. We have molecules of DNA that are surrounded by histones, and histones are going to combine to make what is called the nucleosome. And the nucleosome is going to combine and wrap about to make what is called the chromatin. And chromatin is what is going to make what we know as chromosomes. We probably hear about having the chromosomes associating with each other. So if we compare the picture of before cell division, you see how the chromatin is all around, and then just getting prepared for cell division, we will see that the chromatin combines to make the chromosomes as we know. We know that humans will have 23 pairs of chromosomes, and these chromosomes are bringing the characteristics that we know that can tell us if we're going to have black hair, long legs, or more than that. It contains the information that we know that is the molecule of DNA. If we go back to the same picture, we can say, yes, the molecule of DNA, it will be a double strand. So you can see two strands of the DNA that they are surrounded by one another. That is made out of genes. And in a gene, that will be a segment, a piece of the DNA that is going to be the code for the synthesis of one protein. And that molecule of DNA is so long that it's going to be around these proteins that we call histones folded to make what is called the nucleosome. Nucleosome forming what we call the chromatin. Chromatin, and this will be the nucleosome. But we discuss a little more of that because that is going to bring the stability of the molecule of DNA. Why is so important the stability of the molecule of DNA in the nucleus? It is to protect that information so that it cannot be damaged. So we're going to see, before we look at the properties of molecular DNA, how replication works, and how transcription and translation, we need to understand what are the components. So before looking at the components, we're coming back to say that this molecular DNA will be very stable inside the nucleus because it's packed in the nucleosomes. How is that working? Since we have polymer of nucleotides with the phosphodiester bonds between the sugar units. So these are deoxyribose. We're going to look at that in a minute. Ribose. And 2-deoxyriboses. And then you will have a phosphate unit in between with negative charges. That negative charge is going to be neutralized by the lysine amino acid that is contained in those proteins. So we're going to say that these proteins called histones are very rich in lysines, and also that this negative charge is going to be stabilized by magnesium 2+. We say always that magnesium 2+ is the counter ion of phosphate units. So that is going to be important to make a molecule of DNA that is so long because it's 3 meters long, and then it's going to fit inside the cell because of the wrapping about all of these proteins. And then we're going to say what are the functions that we can look at as just an introduction. So we said in the nucleus we have the molecule of DNA wrapped about histones, making nucleosome, making the chromosome. Before cell division, that molecule of DNA is going to replicate, and the daughter cells is going to have the same number of DNA molecules as the parent DNA molecule. So we will have that the molecule of DNA will not exit the nucleus, but it's going to send the information so that the protein can be made by the ribosome using 3 different kinds of RNA molecules. So that is one of the most important functions of nucleic acids. But we also should not forget that we have, besides DNA and RNA, we also have coenzymes. In the chapter of enzymes, we look at having those organic molecules that are important to help the enzyme in the active site. And in this, I can present just a few here. We have FAD is a flavine adenine dinucleotide, so it is also a nucleic acid. The ATP molecule that is required for many reactions that are endergonic to become exergonic is also a nucleic acid. NAD+, that is an electron carrier, important for the metabolism of many substances. We will need to use this important coenzyme. So it's not limited to have the information that we can determine what is the trait of a person or what is the protein to be synthesized, but as part of the function of nucleic acids is many parts that will contain an ADP part or other different nitrogen base. So if coming back is just remembering that no matter what is the species that we look for, all the components will be the same for the molecule of DNA. In all cases, we will have a nitrogen base, a sugar unit, and a phosphate. Let me repeat. In all different kind of species we will have, a horse will have 32 chromosomes, 32 pairs of chromosomes. The mosquito will have 3 pairs of chromosomes. Humans will have 23 pairs of chromosomes. But in all cases, the molecule of DNA will be made from the same component, a sugar unit, deoxyribose, a nitrogen base, and a phosphate unit.